Gene exchange can occur between microorganisms by a variety of means, including one called conjugation. Conjugation requires cell-cell contact, typically initiated by a special sex pilus protruding from a donor cell and contacting a recipient cell, as shown here with E. coli cells. Sex pili grow and shrink as they sample the environment around them. The tip of the sex pilus attaches to a receptor on a recipient cell, and then the pilus retracts, drawing the two cells closer. The donor cell is called an F+ cell because it carries a plasmid called an F factor, for fertility factor. The F factor contains all the genes needed for sex pilus formation and for DNA export. The recipient cell lacks this plasmid and is called F-. Both cells have a normal bacterial chromosome. The F+ cell makes a variety of proteins involved in DNA transfer, including a large multi-protein complex called a relaxosome at the base of the pilus. Conjugation begins with cell-cell contact between an F+ cell and an F- cell. The membranes fuse, forming a membrane bridge. The plasmid encodes a number of transfer or TRA genes, whose protein products carry out the transfer of DNA. The F factor also contains a site called ORI-T for origin of transfer, where the transfer of F factor DNA begins. TRA-I, also called relaxase, works in combination with other F factor encoded proteins to form the relaxosome complex. Here, the relaxosome is shown wrapped in F factor DNA at a membrane bridge. The relaxase enzyme nicks the phosphodiester backbone at ORI-T. The nick produces a 5' and a 3' end on one of the DNA strands. The relaxosome remains bound to the 5' end of the nicked strand, and the DNA protein complex is transferred through a pore protein into the recipient, where it remains associated with the membrane while the rest of the strand is transferred through the pore. As the DNA is being transferred, DNA polymerase in the donor synthesizes DNA, replacing the transferred copy. In the recipient, a second strand is formed by lagging strand DNA synthesis, which requires the synthesis of RNA primers that DNA polymerase can use as starting points. The RNA primers are later replaced by DNA. When the transfer is complete, the relaxase enzyme ligates the 5' end it was holding to the 3' tail of the transferred strand. Thus, the last portion of F factor that is moved to the recipient is ORI-T. This transfer process is very quick, taking less than 5 minutes to transfer the entire 110 kilobase F factor. Ultimately, the conjugation complex spontaneously comes apart and the membranes seal. The F- cell has been converted to an F+ cell that is now capable of conjugating with another F- recipient. Consider that if the plasmid carries a gene for antibiotic resistance, the resistance trait can quickly spread through a bacterial population.